Hello everyone. In Metabolism of Proteins, we are going to discuss today about the metabolism of the simplest amino acid, which is glycine. So glycine, as you know, it has a structure which is very simple. That is, you have a COOH group, that is an amino acid group. Okay, you have an amino group, an acid group. One side you have H and the other side also you have H. So it does not have an asymmetric carbon atom and it has no optical activity, and this is the simplest amino acid. Now, based upon its metabolic fate, it comes under the group of glucogenic amino acids, and based upon the nutritional requirement, it comes under the classification of non-essential amino acids. So it can be synthesized in the human body from other amino acids. Now let us see what are the steps in the synthesis of glycine. Okay, the first one, serine. Our amino acid OH containing amino acid serine can synthesize glycine by using the serine hydroxy methyl transferase. Okay, this is a methyl transferase and it is a one carbon metabolism based reaction. Okay, so serine is converted to glycine, and another amino acid can also be converted to glycine, that is 309, by the enzyme 309 aldolase. You get glycine. And also there is another enzyme which can synthesize glycine, that is glycine synthase. So if you have only carbon dioxide and ammonia, you can generate glycine. So that is by the enzyme glycine synthase. So there are three steps in the synthesis of glycine. Now coming to the breakdown of glycine. So the chief enzyme for breakdown of glycine is glycine cleavage system. Okay, glycine cleavage system, referred to as GCS. It is a multi-enzyme complex. There are three enzyme activities within this enzyme. Okay, now let us see how that takes place. So here you have your glycine. Okay, so first glycine is decarboxylated. That is, you remove a carbon dioxide. So this enzyme is decarboxylase. Okay, you have the enzyme number 1 is decarboxylase. Glycine decarboxylase. Okay, now once carbon dioxide is removed, this particular multi-enzyme complex has lipoic acid in it. Okay, lipoic acid is a vitamin form, and this lipoic acid is attached to a H protein which is there in the center. Okay, now the remaining part of glycine after the carbon dioxide is removed goes through this, goes attached to this lipoic acid, and then you have the amino methyl transferase reaction. Okay, amino methyl transferase reaction, which is again a folate dependent reaction. It is again a one carbon metabolism based reaction, and now this again goes through this again the lipoamide is modified and the lipoamide needs to be regenerated. So you have the enzyme lipoamide dehydrogenase here, lipoamide dehydrogenase. Okay, so this is a NAD dependent enzyme. So lipoamide dehydrogenase, decarboxylase, and here you had your amino methyl transferase. So these are the three enzyme activities which are present in our glycine cleavage system. So as a result, if you see, you have removed carbon dioxide, you attach the amino group here, and the ammonia is removed separately during the methyl transferase reaction, and the protein regenerates back to its original form via the lipoamide dehydrogenase. The enzyme comes back to its original form. So this is a multi-enzyme complex, and this catalyzes the breakdown of glycine. Now, glycine is very important because it has numerous special products and they have much biological significance. Now, let us see the various products derived from glycine. The first one is creatine. You need to know the details about creatine, how it gets synthesized and what is its fate and everything. So that we'll be seeing next. And the other one is glutathione. You remember we have seen it in cysteine metabolism, glutathione. Gamma glutamyl, cysteine, glycine. Glutamic acid, cysteine and glycine. So it comes under cysteine metabolism. It comes here under glycine metabolism also. And then heme. Heme, which is a part of hemoglobin, myoglobin, cytochromes, and it is a tetrapyrrole ring. That heme is also synthesized from glycine in the body. Then you have the purines, adenine and guanine necessary for DNA and RNA, the C4, C5 and N7 atoms. This you need to know because it's a MCQ question. Okay, C4, C5 and N7. Purine ring has 9 atoms. Out of the 3 atoms are contributed by glycine. So these are the various special products derived from glycine. Now, there are other functions of glycine as such. It is an inhibitory neurotransmitter. You remember glutamate, right? Glutamate is an inhibitory neurotransmitter. Glycine is also an inhibitory neurotransmitter. Then we have seen glycine helps in the conjugation of bile acids. Glycine and taurine helps in the conjugation of bile acids. And also, it helps in the detoxification of benzoate. Detoxification of benzoate to form hippuric acid. So detoxification of benzoate to hippuric acid. This also requires glycine. So these are the various functions of glycine in the body. Now let us have an overview of the complete glycine metabolism. So see here. You have your glycine here. Okay. So glycine, if you see, it can be synthesized from serine by the enzymes serine hydroxymethyltransferase. Okay, and this serine can come from your glycolysis phosphoglyceric acid through various steps, glutamate, and you have serine. Okay, so this is how glucose is linked to glycine metabolism. Glycine is synthesized, and this glycine has numerous functions. I told you purine bases, right? It is involved in the synthesis of purine bases. It is involved in various methyl transfer reactions, and these formate. Okay, all these are related to this one carbon metabolism. And apart from that, if you see, this glycine can be broken down into ammonia and carbon dioxide. For this, you need the enzyme glycine cleavage system. And there are various special products. If you see, conjugation of bile acids, the purine bases, creatine, glyoxylate, glutathione, porphyrins. So as a result, because heme synthesis requires hemoglobin, myoglobin, okay, myoglobin and various cytochromes. All these are the special product pathways. And apart from this, glycine can form components of various proteins. And the most important one is collagen. Collagen is the most important protein of the extracellular matrix, and this is made up of repeats of glycine XY amino acid. And the X and Y will be commonly proline and lysine. Okay, hydroxyproline and hydroxylysine. And collagen is responsible for the tensile strength of various tissues. So collagen is also one of the special products which is derived from glycine. This gives an overview of glycine metabolism. Now let us see in detail about creatine and its synthesis from glycine. Okay, so the first step, how it gets synthesized. You know what is creatine? It is a tripeptide. Okay, tripeptide, that is 3 amino acids are there. What are the 3 amino acids is what we are going to see. Arginine and glycine are the first two amino acids. Arginine and glycine. Okay, these two together will combine and form a compound guanidinoacetic acid. Guanidinoacetic acid. Okay, and it releases ornithine. The enzyme is amidinotransferase. Amidinotransferase, and this step occurs in the kidneys. Okay, this is the first step of creatine synthesis. And you have 2 amino acids which are involved. Glycine and arginine have come inside. Now the third amino acid, which is methionine. I told you already, methionine cannot come as such. It always performs its function in an activated mode, that is S-adenosyl methionine. Okay, so methionine will be converted to S-adenosyl methionine, and it combines with this guanidinoacetic acid to form creatine. And the enzyme is guanidinoacetate methyltransferase. So we have seen all the transmethylation reactions, right? So S-adenosyl methionine, after this methyltransferase, it becomes S-adenosyl homocysteine. So this is one of the transmethylation reactions, and this step occurs in the liver. Okay, so these are the 2 steps of synthesis of creatine. So what is this creatine used for in the human body? Okay, creatine will be phosphorylated by a kinase enzyme, creatine kinase, to form creatine phosphate. Okay, creatine phosphate is an immediate source of energy for muscle contraction. It is an immediate source of energy for muscle contraction, and this is synthesized by creatine kinase. This creatine kinase reaction is otherwise referred to as Lohmann reaction. Lohmann's reaction. Okay, so creatine to creatine phosphate by creatine kinase, and this creatine kinase has much of clinical relevance. You have 3 isoenzymes of creatine kinase, the CK, MB, MM and BB. Okay, out of this CKMB is important because it is used in the diagnosis of myocardial infarction. Okay, so that is the significance of this creatine kinase enzyme. And that creatine kinase enzyme is used for the synthesis of creatine phosphate. Now we have produced creatine phosphate. It is used for energy for muscle contraction. Now this creatine, once its function is over, it needs to get excreted. How does that occur? So once your creatine, its function is over, they lose a water molecule. Water is removed and cyclicization of the structure occurs and it forms creatinine. So creatinine is nothing but it is a cyclic anhydride form of creatine. Cyclic anhydride form of creatine. And this gets excreted in the urine. This is the excretory form of creatine. And creatinine is a very important marker of renal function. You know, creatinine is used for studying the clearance and it is an indicator of glomerular filtration rate. So creatinine. So this pathway of creatine phosphate has a lot of clinical relevance. Okay. Creatine is a source of energy for muscle contraction in the form of creatine phosphate. It is a high energy phosphate. Okay, and then you have the CK reaction, which is a creatine kinase reaction, which has clinical relevance. It is a marker used for muscular dystrophies and also in myocardial infarction. And in addition, you have the creatinine, which is an indicator of renal function. And it thereby gives an estimate of glomerular filtration rate. Now let us see about glutathione, which we have already seen under cysteine metabolism. It is a tripeptide made up of gamma glutamyl cysteine glycine and it has various important roles. That is, it is a free radical scavenger. It maintains the RBC membrane integrity along with NADPH. It has a role in the absorption of amino acids via the Meister cycle and it is involved in conjugation reactions, that is phase 2 reactions of xenobiotic metabolism. These are the significant functions of glutathione in the human body. Then you have the next special compound, which is heme synthesis. Heme synthesis is again a very huge pathway, but here we will see the step where glycine is used in heme synthesis. So if you see the first step of heme synthesis is glycine and succinyl-CoA will join to form something known as ALA. Aminolevulinic acid. These steps in detail will be discussed under heme metabolism. So the first step of heme synthesis, glycine plus succinyl-CoA will give ALA, aminolevulinic acid, and series of steps will continue to form heme, which is a tetrapyrrole ring and it is a component of myoglobin, hemoglobin and the various cytochromes. Now also we saw various purine atoms. Atoms of the purine ring are also derived from glycine, right? So this is the structure of purine. You have 9 atoms in purine ring and if you see, glycine will contribute to C4, C5 and N7. All the other atoms are contributed by different compounds, which we will see in detail in our nucleotide metabolism. Here you just remember glycine will give rise to C4, C5 and N7 atoms of a purine ring. Now let us see disorders of glycine metabolism. There are 2 major disorders. One is non-ketotic hyperglycinemia. This is because of the deficiency of glycine cleavage system. So glycine cleavage system is an enzyme which is going to break your glycine. So what if it is going to be defective? You have increased levels of glycine. Okay, increased levels of glycine in the blood, it gets excreted in the urine and various other body fluids. And glycine also acts as an inhibitory neurotransmitter, so it leads to the development of neurological manifestations. This is non-ketotic hyperglycinemia. Then you have one other disorder, which is primary hyperoxaluria. This is because of the deficiency of alanine glyoxylate aminotransferase. Okay, alanine glyoxylate aminotransferase, and this leads to the production of oxalate stones. Oxalate stones in the urine. Primary hyperoxaluria. This leads to urolithiasis, nephrolithiasis, that is a development of stones in the urine, and the stones are oxalate stones. Primary hyperoxaluria. It is a protein targeting defect. A protein which is supposed to go to the peroxisomes does not go to the peroxisomes and it lands somewhere else. So it leads to different alternate pathways where you synthesize glyoxylate which can get converted into oxalate, and this gets deposited as stones and gets excreted in the urine also. So these are the two disorders of glycine metabolism. So we have seen about what is glycine, from where all you synthesize glycine, and how is glycine being broken down, the various special products, more importantly about creatine in detail, and then we have seen two disorders related to glycine metabolism.